all know about, or we are familiar with bacterial pathogens and infectious diseases, right? So inside bacteria, there are secret RNA conversations happening during infection or during infection. And today, I'll show you how to spy on those RNA conversations, uncover them, and why that matters. So before we move forward, we are going to do a quick refresher about the basics. So first, in bacteria, gene is a piece of DNA which carries instruction. And the DNA is copied into RNA, particularly messenger RNA. So the DNA gives the message and the RNA carries it. And then RNA make proteins which do the job, actual job in a cell, based on the given message. But there's an additional layer which is called small regulatory RNA. So these bind to mRNA and control whether the proteins get made or not. So I mentioned in the beginning that we are going to spy on these RNA conversations, right? So why should we care about these RNA conversations in bacteria? So when bacteria enter a host, for example us, to cause a disease, they need to rapidly adapt to a hostile environment by turning the genes on and off to progress the infection or to survive. So major part of this control comes from this small RNA. So these tiny molecules control the gene expression very rapidly and quickly and efficiently by interacting with the target messenger RNA. So if we understand which small RNA is talking to which messenger RNA, we can find ways to control the infection. So as I mentioned already, these inside bacteria, the RNAs are constantly conversing. So it is like a crowded room with full of whispers. We know conversations are happening, but we don't know we can't tell about who is talking to whom and what they are talking about. Likewise, in bacteria, we didn't know all the interactions happening inside the bacterial cell, and we wanted to know which small RNA is talking to which mRNA. So all the methods so far looked into like one or couple of interactions at a time. So we are kind of missing the whole picture of interactions as well. So this is where an intuitive methodology came to play, which is called RILSEQ, RNA interaction by ligation and sequencing, to find the real interacting pairs. So in this methodology, there are three key players. The first one is mRNA, as I mentioned already. This is a message which has to make proteins. And small RNA, it is a controller that binds to mRNA and regulates it. And finally, the third one is the protein, HFQ, which is a matchmaking protein, bring these two RNAs together. So the first step, we freeze these RNA and protein interaction in place. And secondly, we link these interacting pairs physically together. And finally, we read these pairs that are actually talking by using sequencing. So what do we learn from here? Instead of getting single interaction at a time, we get hundreds of interaction. And we observe like one small RNA is controlling multiple mRNAs, or more than one small RNA is controlling a single mRNA. So we are kind of moving from single interactions to a full network of regulatory interactions, and how cool is that? So we performed this strategic mapping for the very first time in a bacterial pathogen called Brucella abortus, which cause abortions in farm animals and debilitating fever in human. Unfortunately, we didn't have like antibiotic treatment to control the infection in humans. So again, why my findings matter? So by hearing these secret conversations and understanding the complex interactions during infection, we can find the weak points in the bacterial system. By weak point, I mean, as I mentioned already, one small RNA is controlling multiple mRNA, right? So this one single small RNA could be a weak point. And by interfering with these weak points, we can find ways to control the infection, and also we can use this information to develop some therapeutic measures. And yeah, that's it. Thank you.